Hello, everyone, and welcome to today's live broadcast, Fast and Easy Identification of Disease-Causing Variants in Hereditary Diseases Using Patient Phenotype Information and Testing Different Modes of Inheritance, presented by Dr. Anika Joeker. We are excited to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. This webinar is sponsored by QIAGEN Bioinformatics. QIAGEN Bioinformatics is powered by Bio, Ingenuity, and Biobase. The company offers bioinformatics software tools for next-generation sequencing, NGS, data analysis, and interpretation. Its solutions are designed to be universal so customers can mix and match technologies best suited to their needs. For information, please visit QIAGENBioinformatics.com. I am Christina Mahalik of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type the question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you'll be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing this presentation, please click on the support button at the top right of the presentation window or use the Q&A button to let us know that you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page and follow the process of obtaining your credits. Now I'd like to introduce Dr. Anika Joeker. Anika Joeker is Director in the Global Product Management Department in the clinical program of QIAGEN Bioinformatics. She's been working in the field of bioinformatics for the last 15 years and worked as bioinformatics group leader at the German Cancer Research Center before she joined CLC Bio, now QIAGEN, in 2011. Please join me in welcoming Dr. Joeker. I will now turn the presentation over to her. Doctor?
Thank you very much, Christine, for the introduction. In the next about 45 minutes, I will guide you on how to analyze or how to find the causal mutation from a trio. So that means mother, father, and child. So the outline of my presentation is that I will give you a very short introduction to QIAGEN Bioinformatics. And then afterwards, I will talk about the challenges when you analyze trios to identify the cause for hereditary diseases. Then I will proceed by talking about QIAGEN's solution for hereditary diseases. We call it QIAGEN's Hereditary Disease Solution. And in the end, I will spend most of my time in showing you 3 examples. One is de novo mutation, the other one is dominant inherited, and the third one is homozygote recessive. QIAGEN Bioinformatics likes to bring a software solution for all needs of a genetic testing lab. So from the discovery of new findings, which often occurs in academia, to the testing of patients for specific mutations, but also, and that is mainly what we talk about today, the advanced testing part where you actually back like in a research setting and you investigate what could cause a specific disease. So it's a little bit like finding a needle in a haystack because especially when you look at rare inherited diseases, rare diseases in general, you don't know from the beginning what you look at. Therefore, a whole exome or whole genome sequencing is the right way to go. QIAGEN bioinformatics is a relatively young subunit of QIAGEN. So QIAGEN is well known as a leader for sample extraction kits, so to extract DNA from different kinds of tissue. In 2013, QIAGEN bought 3 bioinformatics companies. CLC Bio, which was famous for data analysis tools and software, Ingenuity Systems for interpretation software, and BioBase for biological databases. I'm sure you know some of our products, for example, the HEMD database, which is currently the gold standard in the field for annotating of variants in hereditary diseases. The other tool you also might know is Ingenuity pathway analysis for pathway interpretation, or the CLC Genomics Workbench for data analysis. So these 3 companies don't exist on its own anymore. So we are now all sitting in one new unit of QIAGEN. And also when we are relatively young as part of QIAGEN, we actually have quite a lot of experience. And therefore, QIAGEN became overnight one of the leaders in the bioinformatics space. So we are currently hosting one of the biggest knowledge bases which you can find in the market, so which has over 13 million biological findings, and many of them are human-created from literature. So we have up to 200 people sitting and reading literature on a daily basis and incorporating that in our knowledge base. On top of that, from this literature creation, we also have integrated open source and commercial available databases like Cosmic, then our own database, HEMD, ClinVar, and several others. So in total, our products are cited in over 30,000 publications. And this number of sample analyzers is actually pretty old, because this is actually just one of the software, and we have analyzed already now over 420,000 samples. In total, in our unit are sitting close to 350 people working on bioinformatics, and that shows you that QIAGEN is not a small company for offering bioinformatics software. So one of the big highlights of our software and software solutions is our knowledge base. And in the beginning, BioBase and Ingenuity and now QIAGEN has built up this really huge knowledge base with over 13 million biological findings by doing literature curation. But of course, you can just, it's easy to read literature and put all the knowledge into a database. But this makes only sense, or it's only useful, if you build an ontology with it. That means you build a connection between the terms, which allows you to use the information as part of our software by building connections. So for example, if a variant in a specific gene is known, and the gene is known with a specific disease, or the gene is interaction in a pathway, and this pathway is known with the disease. So this kind of relationship you can do if you organize all your knowledge in an ontology. So of course, when you would put just random people, we would just choose them, and then they would put random stuff into the knowledge base. It would also not be very useful. Therefore, all our people are trained and certified on how to read and create the literature. And then there's also a very high quality standard in the knowledge base. So we have reviewers and people who are constantly checking the quality of this database. And in the end, our software, so our interpretation software, QIAGEN Clinical Insight, Interpret, and Ingenuity Variant Analysis, as well as QIAGEN, as well as Ingenuity Pathway Analysis, have access to this knowledge base and can use the information. So we are talking today about trio analysis. So what are actually the challenges when you analyze or when you try to identify the cause for a disease, for an inherited disease, from a trio? That means you have father, mother, and child. So first, why is it actually beneficial to have father, mother, and child available? That is because you can use, in case, for example, the proband has a disease, but the mother and father don't have any symptoms, you can use them as controls. And when you do tree analysis, so this is an easy case for de novo variants, you think, then you do variant calling in the mother, in the father, and in the sample of the proband. And then, so you're not interested in any variants that are present in the mother or in the father, at least not directly. So when you look for a de novo variant, so de novo means it's just happening in the child. So you would remove all the variants which you identify in the mother and in the father from the list you have found in the proband. And that would be your candidate list of variants. The problem is when you do this approach, you can easily end up with thousands of variants. Why is that the case? For sure, they're not all de novo because normally we are expecting between 4 and 10 de novo variants in one of the samples. So why is there so many of them? So they're almost all false positives. And that is because you have changing coverage, read coverage in the samples. So you could, for example, have a very high coverage in one of the parents, but a very low one in the child, or the other way around. That could also be, and that is actually pretty unknown to many people, that one of the alleles is not very good enriched in one of the samples. So that means that when you have, for example, this is a de novo variant in the child, but this variant is actually also, so you have found something in the child, but it should also be present in the parent, say in the father. Then in the father, maybe that allele wasn't enriched really well, so it was maybe at a frequency of 1% of the sequencing reads. So you might miss those when you do variant calling. So that means you have to spend a lot of time and your own resources to ruling out false positives at this point. Of course, some people also use now a joint calling. So they are doing a read mapping of mother, father, and child together and calling the variants from all of the samples together. But also in this case, you might end up with a pretty huge list. And de novo variant is actually the easiest one. Let's say you have dominant variants or recessive variants where, so in the dominant case, for example, you have just one of the parents which you can actually remove variants from because he or she is unaffected. Yeah, the problem is, I'm pretty sure for those of you working on hereditary diseases that you have had that before. So currently, the case-off rate for hereditary and rare diseases is around 22 to 30%. That means in 70% of the cases, you cannot find a disease-causing variant. So what are reasons for that? So if you haven't sequenced, so if the variant was not in the target region you have sequenced, of course, you cannot identify it. Then also the best bioinformatics software cannot help you. And there could also be other reasons for the disease. So, for example, mutilation. But it could also be that the variant was missed during the variant calling because there was not enough coverage at this position or because of this allelic dropout. And you can see that on the screen here, this is an example. In the mother, you see a variant pretty clearly. So and it is still in the minority of the reads, but it was detected, a G to an A change. In the proband, you also can see it, but it wasn't caused because there are just 2 reads which are supporting the variant. So you might miss that in the proband, and let's imagine that this is a dominantly inherited variant. So that means you wouldn't call it as such. But it could also be that the variant is actually unknown to be associated with a specific disease. So the one problem is to call it, but the other one is to interpret it. And what most people do in that case, they use open source database like LINVA, for example, or others to annotate the variants with known variants from those databases. But if the variant is not present in those databases, you might not recognize it as in this case, pathogenic or disease-causing. So that means for you, because you cannot find the cause, either you cannot help the patient at all, or you cannot, so you cannot report something back. So that means, or you do more experiments. So for example, from a whole exome sequencing experiments, go to a whole genome, or do other kinds of assays to find a good candidate. But that would also mean higher costs. The third challenge is something which many people are actually maybe not aware of. And that is that you can have the different software pieces doing different kind of analysis, and they are not good working together, also when you think they are. So software A, for example, is often used then for the variant calling. So in many cases, GATK is used for that. Software B for the data interpretation is then either a self-built filtering pipeline or a notation pipeline or a commercial software piece. And software C for data visualization, most people use the integrated genome viewer. The problem is that per se, all these tools are not working really great together as one pipeline. So that means you first need someone who builds up the pipeline to automate it, so that when you have many samples, so you can run them all together. The second one, so that means you first need to hire someone. Then also this person has to maintain, which is not always the fun part. So if you want to have small changes there, he or she becomes maybe a bottleneck. But then the other problem is also that not all software pieces provide an API you can program against. That is mainly software B for data interpretation. So that means that in the worst case, the whole thing becomes very manual process. So you call variants of software A, then you get out the VCF, and then you have to manually import that in software B to have it interpreted, and then you end up with a BEM and the filtered and annotated variants you have to load then in software C to confirm that the variant is really present and not a false positive. And that, of course, is not doable if you have a lot of cases. QIAGEN is bringing to the table here its own hereditary disease solution, which is built out of 3 components. These 3 components can be used independently, but if you use them together, they work like one product. And that is Biomedical Genomics Workbench and Biomedical Genomics Server Solution. Both is a software you install locally behind your firewall, and then they can identify variants, and they upload the files then to Ingenuity variant analysis, where they get interpreted and filtered by using this very huge knowledge base that QIAGEN has. Then the visualization is happening in Biomedical Genomics Workbench in an interactive genome browser, where you can validate your results. So you can jump back and forth between the software pieces. And when you are happy, then you can report them into QIAGEN Clinical Insight Interpret, where they get also classified in pathogenic and benign, and you get a clinical report out. So this is a software solution which enables not just the variant calling, but also the filtering, the data interpretation, and the visualization of results, and the reporting in one product. And this is the only one which currently exists like that in the market. So you start with your FASTQ file. So directly from the sequencer, you load in your FASTQ files into Biomedical Genomics Workbench, and then you can do, if you want, some QC reporting there. So to look, for example, is there any adapters hanging on the sequencing reads, if the enrichment was good, and so on. The next thing you have to do is just to run one workflow. So that's a one-step workflow. You double-click, and then you can change some of the settings, not all of them, because that's not necessary, because the workflow is already optimized. And this workflow not just does the variant calling, but also the filtering, the interpretation, and even the visualization in one step. So what you get back out of the workflow is a genome browser view with a read mapping from the mother, father, and the proband, overlapping with the genome regions, and what you also get is the results, the filtered, so a candidate list of variants coming back from Ingenuity variant analysis. But at this point, you should not directly look into the visualization here and confirm the results, because there is an essential thing missing here. And that is that you should add phenotype information. about your patient. So you go into the genome browser view in Biomedical Genomics Workbench. You right-click on the track, which comes back from Ingenuity variant analysis, and then you say launch Ingenuity variant analysis in browser. So that opens up then the filter cascade, which was one in Ingenuity variant analysis, which does different kinds of filtering. So for example, it filters out variants which are common in the population. It filters out variants, so it keeps variants which are pathogenic or likely pathogenic, are present in HGMD or different other criteria. And then also it does the filtering for the inheritance pattern, for a specific inheritance pattern. But minimally, so you should add another filter, and that is a new filter we have, which is called phenotype-driven ranking. So what this filter does, so you provide it with the phenotype information, so as many phenotypes as you have for the patient, and then this filter does a ranking, a prioritization of the variants based on the phenotypes. So all variants which are best explaining most of the phenotypes are ranked the highest, and the ones which are maybe just explaining one of the phenotypes the lowest. And after you have adapted, of course, you can also adapt the other filter settings. You go back into Biomedical Genomics Workbench, you right-click again, and you say Variant Analysis Update. And it will then fetch the latest result from the filter cascade, and import that into Biomedical Genomics Workbench, where you then interactively validate and inspect them in a genome browser view. But of course, before you validate, you want to make sure that you really have a really good candidate here. And there we come to the inspection of variants, and that you can do, the best is to do that in Ingenuity variant analysis. So at the end of the filter cascade, you get a list of candidates. And the list of candidates you can is maybe then ranked because you have the phenotype-driven ranking filter applied. But you can also, you should actually look at the publications or external literature information for each of the variants. So, for example, you get information if the variant was identified before in any of the databases or in the literature described. So if it was described in the literature, you get the link to the literature, but also a sentence which summarizes in what context the variant was identified, and with what kind of disease, if there's any. You also see if it was what the functional impact could be. You see also a classification here, and you get information from population databases, how frequent it is in the population. But what is most useful is this genotype to phenotype relationship. And that is directly from our knowledge base. So let's say, for example, you have a variant which is in a gene, and the gene is not directly associated with the phenotypes. Then you might miss those. Our software allows you to have a step in between. So for example, if the gene is known in the pathway or is interacting with a range of proteins which are then associated with the phenotype, then you are able to spot those and get these in the end list. At the end, if you are happy with whatever candidates you have filtered at the end for different inheritance patterns, then you can validate them in Biomedical Genomics Workbench together with map sequencing reads and then confirm or reject. Because it can still be that at this point you have a really great candidate, but you find out that this is actually a false positive. And when you have confirmed and you're really happy, then you can press a button in Ingenuity variant analysis and directly send the results to QIAGEN Clinical Insight Interpret, where a report is written out with the causal variant. Causal variants you like to report. You can also add comments. You can add literature information a classification, and so on. And this report can be then handed out to a physician. But what makes this workflow actually pretty unique? These are mainly two things. So it does not just do variant calling, mother, father, child, and then remove the variants from mother or father, depending on what kind of inheritance pattern you look for. It also does something more fancy to make sure that also when you have low coverage or allelic dropout, that you pick actually all disease-causing or potential disease-causing variants up. So how does it do that? So it calls variants in mother, father, and child, so that's the same. But then it combines the variants from mother, father, and child to a big variant list, and it does a checking back in the map sequencing reads of any of the individuals to see, if, for example, if you have found a variant in the proband, if that is not missed in the reads or in the variant calls from the mother and the father. So that means, for example, in the case we had beforehand, if you have called a variant in the mother and you haven't called a variant in the proband, then this backcheck allows you to also identify it in the proband also when it's just present in or supported by two sequencing reads. And therefore, you get an enhanced list of variants going back, and these are then uploaded to Ingenuity variant analysis for filtering and interpretation. But the workflow does also a little bit more. So another challenge is when you'd like to find the cause for, let's say, a rare disease. So your parents, for example, don't have any symptoms but the proband. Then you often need to test different inheritance modes. So, for example, it could be that this is caused by a Homozygote recessive mutation, which the child has inherited one allele from the father and one from the mother, and that was the same allele. So in total, the child becomes homozygote. And that will cause a disease. Or it's de novo, so it just happened in the child. But it could also be that it's compound heterozygote, so you inherit one from the mother, from the father. But this is not the same variant, but it's in the same gene at different location. And this is causing then the disease. But it could also be that this is actually caused by a dominant variant, and that one of the parents was affected as well, but you might not have seen it, or the physician has not seen it. That often happens, for example, for intellectual disability, if one of the father or the mother has just mild symptoms. The workflow is able to test all four inheritance modes at once. So you get a list of dominantly inherited variants, homozygote variants, compound het variants, and de novo variants. So we have benchmarked the workflow also and on one exome and on 6 whole genome trios, which we got provided from the Innova clinic in the US. So with the forward settings, the workflow is able to filter down the variants in case of a whole genome experiment from close to from 6-7 million variants to 20. In the worst case, it was 266 variants. And when you add phenotype information, you can see that in the worst case, and that was a dominantly inherited variant, we end up with 30 candidates. And that is something where you can really manually go through this list. So now we're coming to the examples. So in example 1, we have a mother, a father, both of them are unaffected, and we have an affected proband. So the phenotypes described by the physician is Manning myolokili, unilateral renal agenesis, prominent nose, hirsutism, and retrognathia. So we run the workflow, and it tests automatically all inheritance patterns. So you can see that here. So that is what you get back. So you get the reads for mother, father, child, and then you get 3 variant lists One is the candidates for de novo variants, one for compound het variants, and one for homozygote recessive variants. Normally, you also get the dominantly inherited variants back. Then you can inspect the list and look if you see something interesting. But at that time point, we haven't added any phenotype information yet. So here, a different scenario here. So now we are trying to add phenotype information. So for the de novo case, we had 25 variants. For the compound heterozygote case, we had 4. And for the homozygote case, we also had 4 variants. Now we are adding our phenotype-driven ranking filter. And that was actually just in one case possible to add this filter, because when this filter returns 0 variants, you cannot add it. So when it fails, then you know, oh, this actually returns a 0 to me. So this filter could just be added in the case of the de novo variants. And this filtered down from 25 variants to 6 variants, which were somehow associated with one or several of the phenotypes. So let's have a look at this list of variants. So we have here on top which was ranked the highest and has the highest score. And you can see it's explaining 3 of the 5 phenotypes. And that is a variant in EP300. The other cases, the other variants you can see here are only explaining 2 of the phenotypes or just 1. So they are not really good candidates. So we inspect now the variant in EP300. So if you press it, you also get a link. So you see here the gene EP300, and you see that this is supported by literature to be associated with Rubenstein 2B syndrome type 2. And this syndrome is then associated with the 3 of the 5 phenotypes. So this is a very strong candidate, and when you click on one of the links, you also can see the underlying literature which is supporting it. It was also automatically classified as pathogenic for this type of disease. When we then went back into Biomedical Genomics Workbench and we update the results so that we just see the 6 variants and we look for the one from EP300, then we can see that it's just present in the proband and not in the parent. And we can also see that the quality of the reads looks really good. So they're all in blue, which means that they are mapped unambiguously. So there was just one position where these, so it was not a homopolymer region or anything. You can also see that the reads were aligned to each other because they are blue in a distance which was expected. So overall, this is a really great candidate, and also by just looking into the map sequencing reads, we can validate that this seems to be a true positive and not a false positive. So I have to say that in this case, it was also confirmed. So this is really the disease-causing candidate. Now we are coming to example 2. So you can say de novo variants, that's easy. I have 2 where I can filter back from, but now we're coming to a more challenging case, and that's the dominantly inherited variant. So we have now an affected proband, but also the mother in this case is affected, but the father is not affected. And the phenotypes are heart malformation and malformation of great vessels. So we run again our workflow, and we're ending up with 267 variants by using default settings. And we're assuming here dominant inheritance pattern because the mother is also affected. When we now add the new phenotype-driven ranking filter, then this can be filtered down to 17 variants. And when we look at the 17 variants, then we can see they were ranked already. All of them are explaining both of the phenotypes. However, only the first one was classified as pathogenic. So we look at the first one now to see if how that is explaining the phenotypes. And then we can see that this gene, mu6, was associated with atrial septal detect type 3 and with congenital abnormality of cardiovascular system, and that is then associated with our phenotypes. And that is also supported by the literature. So that is a really great candidate. It's classified as pathogenic, and also in this case, this is actually the one which was causing the disease. When we are now going back into biomedical genomics workbench, we can actually see that this is not a variant which is in a coding region, but in a splice site region. And that is actually makes it even more interesting because many pipelines will miss those because they're just looking at the coding regions. And maybe are even filtering out for non-synonymous variants in there. So you can see the variant clearly in the proband, also in the mother, and it was called. So this is, and you can see that all the reads are mapped in blue, so that means they're in the distance, which was expected, so mapped in pairs. And also there's no homopolymer stretch, they're not yellow, which means they are unambiguously mapped. So this is also something which you can trust. And now we are coming to the last example. And here we have a homozygote recessive variant. So in this case, also the parents are not affected, but the proband is. And for that, of course, you have to test again all 3 inheritance patterns. So the phenotype in that case is decreased fetal movement, seizures, and severe neurocognitive dysfunction. And we run the workflow and we add the phenotype-driven ranking filter. That was actually just possible in the recessive case in that case. So we could really fast see that this is a recessive variant which might cause it. And we can see that the first variant in HSD17B was explaining 2 of the 3 phenotypes. And that was the only one which was recessive. You inherit 1 from the mother and 1 from the father. And therefore, it also has a higher score. So that is our strongest phenotype. And when we look at the genotype-phenotype visualization, then we can see that this gene is associated with different kinds of syndromes. So one is gonadal dysgenesis with auditory dysfunction, autosomal recessive inheritance. This is exactly what we have. We have an autosomal recessive inheritance which cause movement disorders, cognitive impairment, reduced fetal movement, and severe cognitive impairment, but also seizures. Yeah, so also in this case, this was a disease-causing variant. And when we look now in the genome browser to confirm or reject if that might be a good candidate, then we can see that this is in the father in a heterozygote state, also in the mother, but in the proband is homozygote. And this is also in a splice region. So if you filter out variants which are in intergenic regions, then this would have been filtered out. So interestingly, this is also a small deletion, but these are often hard also to call. So you might have missed those actually also when you do other variant calling pipelines. So to sum up my talk, so I showed you our solution for Kaizen's hereditary disease solution, which is able to overcome challenges which you might have when you look for the causal disease-causing variant in rare and hereditary diseases. So one of the challenges is that you have slow coverage or allelic dropout, which leads in that case to many false negatives, so you might not actually identify the disease-causing mutation, but also false positives. So some of the variants will show up, let's say, as de novo as candidates, but they're actually not. Then also you have to spend often a lot of time to test different modes of inheritance. That means, in the worst case, you have to manually change the filtering. So our solution does all of this in one step, but it's also at the same time highly flexible and interactive. So it has the benefit that it overcomes allelic dropout and low coverage by checking back in the map sequencing reads to see if really all variants was called in all of the samples. And on top of that, it uses a QIAGEN knowledge base with literature-curated genotype-phenotype relationships, where you can even identify candidates of disease-causing, which I have not directly have a link with a phenotype, but maybe are in the pathway and therefore associated. So what is new in that workflow? So we have this workflow actually for a year now, but what is brand new, and that will be released on the 8th of November, is that it automatically checks for all kinds of modes of inheritance automatically. So you get automatically 4 lists out of candidates for de novo, compound heterozygote variants, homozygote, recessive, and dominantly inherited variants, which reduces the time for you to identify disease-causing candidates to a very, very low fraction. So that means that when you have an exome experiment, already without manual interaction in 5 hours or 3 to 5 hours, you have already results, and the manually part of that consumes, in the worst case, 15 minutes. So for all inheritance modes, the list of candidates is drastically reduced, so you can manually validate them one by one if you want, because the list often contains only candidates which are less than 10 candidates. And this not only works for de novo variants, which are maybe the easiest ones, but also challenging dominantly inherited and recessive cases. So with that, I'd like to end my presentation, and I'm happy to answer all questions you might have. Thank you very much.
No, thank you, Anika, for that informative presentation. It's time for Q&A. If you have a question you'd like to ask Dr. Joeker, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type the question into the box that appears on your screen and click the send button. We'll answer as many of your questions as we have time for. So let's get started. All right. Dr. Joeker, we have, our first question is actually a 2-part question. The first question asks, how quickly can gene identification occur using this technology?
Yeah, so the gene identification can be very quickly. So it depends on the most part, and that runs without interfering or any manual hands-on from you, is the variant calling part. And that depends on the hardware you run the software on. So if you really fast hardware and a very good disk, you can run that extremely fast. If you have a very slow one, then it takes longer. It can also, the software can run even on a cluster. So that means that if you have a trio, among the variant calling can be parallelized. So you run automatically mother, father, and child. And then we're talking here about a runtime, let's say, between 3 and 5 hours, depending also on the coverage, of course.
Okay. And the second part of the question is, how practical is the process in a physician's office for routine use of patient diagnosis?
I think it's really practical because you can, you can run it as it is, so you don't have to optimize it. Of course, here I'm talking about rare diseases, but let's say you're always testing for the same disease, you can actually lock down the pipeline. So you can say, okay, I'm always running the same filter cascade in variant analysis, which you have before optimized. Or you, for example, you always test for dominantly inherited variants. And in that case, you can run many of the samples then on a routine basis with the same filter settings end to end. And that, of course, makes it also easy to validate it.
Excellent, thank you. Let's see, our next question asks, are the literature findings in IVA really variant specific?
Yeah, some of them are. So of course not all literature findings, so that's a fraction. But so some of the literature findings are variant specific and some of them are gene specific. So that means what you see in variant analysis, and can we go back to the, just a moment, I'm going back to the slide so that you can see it. So it's maybe small, but you can see that here under the column variant findings. And just a moment, I will guide you to it, to this column here. This is actually variant findings in databases and from the literature. So that means this is variant specific. The other thing, this genotype to phenotype relationship, which you see here. So this one, that is not variant specific. So that are relationships which are from the gene. So the gene, how the gene is associated with the phenotypes.
Excellent, thank you for that information. Our next question asks, can your trio solution also handle larger insertions and deletions?
Yes, it can handle also larger insertion and deletions. So currently we are able, for deletions actually we have not really a border, so we can, any kind of deletion we can call. For insertions at the moment it's 50% of the read length.
Okay, thank you for that. And once again, we have another, our next question is actually a two-parter. The first question asks, what tools are used to associate genotype and phenotype?
Yeah, so this tool is, so it's a self-built tool in variant analysis, which uses actually the content from the knowledge base. So that means in the knowledge base there's an ontology used, which makes association between genes, phenotypes, genes, diseases, but also gene-gene interactions, and gene pathway information and others. And this ontology is used. So that means when you have a variant in a gene, and this gene might not be directly associated with the phenotype, then the tool itself or the filter can make an associated by using the knowledge base, by using the ontology and say, for example, this gene might be interested, it's in a pathway with another gene, and that gene is associated from the literature with that disease. So in that case, when you use this information, it can make this association, and therefore you can come down to really less than 10 variants in the end as candidates.
Okay, so that may, and then the follow-up question, which I think you just touched on, is how is your list of thousands of variants reduced to less than 10? Yeah, so that's, so this is really how it comes in. So less than 10, there's two things why it can be reduced to less than 10. Number one is the checking back in the sequencing reads, so therefore you can exclude quite a lot of variants, which in one case you would see as candidate, but then by looking back in the sequencing reads, you can actually see that these variants are actually also present in the parents, for example, or in the child. In the child you can see it, but maybe in one of the parents or in both of the parents. And the other part is, and that is also where really you can filter down to such a low number,
is by using the knowledge base with the literature curation and the ontology in it. So you cannot make that if you don't have the knowledge base. Because then you would actually do the same then. You would use like Google Scholar or the NCBI search and then check the variant in what kind of gene this is present, and then you would look what the gene, if that is associated with any other gene. But of course it would be very time consuming. So I have talked with some people who were working on one of the cases for weeks or even months, and to come up with a good candidate.
Excellent. Thank you for that information. Our next question asks, how does the workflow automatically choose the correct genetic scenario?
It does not. So the workflow tests all scenarios. So the workflow tests for dominantly inherited mode of inheritance. It also tests for compound het variants, compound heterozygote mode of inheritance. It tests for de novo case and for homozygote recessive case. So how you come then to the very quickly to your candidate is in the end that you look for all of the different scenarios, and for that we also, let me go back to the slide here. So here you can see that. It runs in parallel all these kinds of scenarios. And you can see that you have in all of the cases, so in the worst case, you have in the homozygote recessive case, so in the worst case, actually the de novo where you have 25. And then you add the phenotype information. So in the phenotype information is not successful in all of the cases here. It was actually just one case where the phenotype-driven ranking filter could have been applied, because in the other cases that would result in a 0, so no candidate. So that means so you get the results for all of the different inheritance patterns. But then when you look in parallel over all of them, you can really quickly come to the disease-causing candidate by adding phenotype information. And then you can automatically reject the other inheritance pattern. Like in this case, for example, we don't know what is causing the disease. So we think it's not dominantly inherited. At least that would be the last one we would choose, because we know that the parents don't have symptoms. So we look for these 3 cases, and for compound heterozygote and homozygote recessive, these 2 scenarios, so the variants here are not explaining any of the phenotypes. So only in the de novo case where the filter could be applied and was reducing it to 6 variants. So and there we found the best candidate then on the top. So so therefore you save a lot of time by doing that in parallel.
Thank you. Thank you for that information. And the questions keep coming in, so let's go to the next one. This question asks, when I have optimized the de novo filter cascade in Ingenuity variant analysis, will I be able to reuse this one on my other trios?
Yes, as I explained before, you can, of course, you can run the workflow as it is, but let's say you have, you always want to test the same scenario, and you want to have so you have a specific, you have changed maybe some of the parameter settings, either on the filtering and interpretation side or for the variant calling, and now you want to apply it on, let's say, 1,000 other trios, then you can make that. So you can lock down the complete workflow, and so not only the variant calling, but also the filter cascade and variant analysis, and then you can run it with exactly the same parameter settings for the rest of the samples, or for the rest of the families.
All right. Okay. Looks like we have time for one last question. This question asks, can I try the solution on my own samples?
Yes, so we have a trial of the software package. So if you're interested, then contact us under www.qiagenbioinformatics.com and or by follow-up email, which you will get. And then you can ask for a trial, and you will get that trial for 2 weeks where you can test it on your own samples. So we have tested that actually already with a lot of customers. And in all cases, we identified the candidate or a strong candidate, and also identified in many cases more what was not seen beforehand. So try it out on your own samples, and I'm happy to hear your feedback.
Excellent, excellent. And with that, do you have any closing comments?
So I have no comments. Thank you for your.
My pleasure, my pleasure. Thank you once again, Dr. Joeker. I would like to thank our sponsor, QIAGEN Bioinformatics, for making today's educational webcast possible. Before I go, I want to let everyone know that today's webcast will be available for on-demand viewing until February 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. And please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again here at LabRoots. Goodbye.